So this is Unit 6, Lesson 3, and our focus is on a process called genetic drift. For the learning intention, you need to understand how the process of genetic drift works, and you also need to understand what is the effect of population size on the outcome of genetic drift. Because depending on the outcome, depending on the size of the population, whether it's very big or very small, there can be a lot of damages done to a population due to this genetic drift process. So we talked about natural selection last lesson, and now we're moving on to genetic drift, where we would define it as a mechanism of evolution in which allele frequency or population changes over generations or over time due to just pure chance, right? So this is like a random chance process. And these changes to allele frequency is due to chance rather than anything else. And let me explain that to you using the simulation right here. So if you click on this link, it's going to take you to this website. Actually, let me do that. Cool. Now, how does genetic drift actually works? Now, if you have a think about an individual, every single individual has a pair of chromosomes, if we are talking about humans or most of the animals or mammals. And because they have a pair of chromosome, for each gene that lies on these pairs of chromosome, they get two alleles, all right? Now, again, we're gonna use gene A as an example. So there are two alleles, a big A and a little a allele. Now, when this generation mate with each other and produce the next generation, they will breed with each other and then they will produce sperms and eggs, and then those are going to get fertilized, and then you give rise to the next generation. However, when sperms and eggs are being made, remember from an individual that carries two different alleles, like the heterozygous individual, half of their sperm or eggs is going to have the big A allele, and the other half would have a little a allele. Depending on just pure chance, so this is what we call a chance process. Depending on the chance itself, whether or not the little a or the big A becomes the egg or the sperm that gets to fertilize and produce the next generation, we're gonna have different allele frequencies to the previous generation. This is based on the assumption that big A and little a give rise to phenotypes or give rise to traits that do not favor them in any way. So both of these traits produced by the big A and a little a allele have equal chance of survival or equal chance of getting a mate in the wildlife. So there is no advantage over each other. This is the major difference between a genetic drift and a natural selection situation. Natural selection, one of these alleles has an advantage over the other one. Whereas in genetic drift, we are saying that none of them has any advantages over the other. It's just by pure chance that the individual somehow inherited this little a from the parents. It's just winning the lottery, basically. So you could potentially by chance produce the next generation that happens to inherit a lot of the little a's instead of the big a's. And then the next generation, you could also by chance inherited the little a's and didn't actually get any of the big a's passed on, not due to any advantages, but due to chance. So your gene pool can change over time or can change over generation due to random chance. And let me demonstrate that to you. So if I click the next button at the bottom, which is right here, It's going to take me to the simulation. Now in the simulation, I could set my population size, I could set the number of generation that I want the population to breed, and I want to set the original allele frequency. So I'm going to be very fair and set it at 0.5, meaning that half of them, 50% of them will be big A, and the other 50% will be little a. And population size, maybe I'll set it to 500. And then number of generations to be run, I think I'm going to say run it for 200 generations. And then I could click plot and it would look like this. Now, this is by random chance that the big A allele somehow decreased over time. So this is generation. This is the frequency of big A allele plotted on the y-axis. I could plot it one more time. So I clicked it one more time. I ran exactly the same experiment with exactly the same values, 500, 200, and 0.5. And I got a completely different result. So the outcome of genetic drift can be quite random. You could suddenly get an increase amount or increase allele frequency, or suddenly get a decrease. I could plot it 100 times, and every single time it's going to generate a very different result, because the inheritance of alleles that do not have advantages over each other is by pure chance. All right. So hopefully you have answered the definition question and answered what is the major differences between natural selection and genetic drift. I want you to think about the nature of the alleles. Are they different? Do they have advantage to each other? I want you to think about whether something's random, by chance, or no, there are selective pressures in it. All right, so pause the video and write those down. Now, if this A allele is having an advantage over the little A allele, that would be a natural selection situation. And what you'll see is it will start off in the middle, 0.5, and then it's just going to shoot up every single time I run it, because the big A could potentially give survival advantages or mating advantages to the individual. However, in genetic drift, that's not the case. So that's the reason why you're going to see a very random fluctuation of allele frequency. Now, I want to show you something a little bit more interesting, is the idea of fixed. So a fixed allele means that it is the only allele left for that particular gene. And I could show you that in the simulation. And let me just plot this one more time. Set it to 0.2. Plot. All right, so you can see that it ran for maybe 10, 20 generations before the allele frequency of big A become completely zero. So from this point onwards, there is no more allele A in this gene pool. And the only allele left is the small a allele. And this is what we mean by fixed. An allele that has become fixed, meaning that it has become the only allele present in the gene pool. Okay, so now we're going to do a little experiment, and I'll let you do that and let you run the simulation. I want you to investigate this research question, which is, what is the relationship between a population size and the effect genetic drift has on the population's allele frequency? So I want you to make a hypothesis first before you do anything. I want you to think about, if I decrease the population size, making it very little amount of individuals in the population, and the number of generation it takes before one of the allele becomes fixed or lost would increase, decrease, or stay the same. I want you to have a think and put your hypothesis down. If you're working with a friend, do it with a friend, have a chat to each other. Next thing is I want you to follow my instruction and run the simulation. So you'll click on the link, which is going to take you to exactly the same simulation. Remember, go to the bottom and click next so you can get to this page that I'm showing right now. Okay, so we are going to keep the number of generation run the same. The population also will keep the allele frequency the same. We'll keep it at 0.5. All right, so keep the 200 and keep the 0.5 the same, but we're going to change the population size every single time we run it. So the first one I want you to do is do 20. So set the population size to 20. And I want you to click plot 5 times without clearing it. And you're going to have a graph generated on the right-hand side. I want you to click it 5 times, and then I want you to screenshot your graph and put it in here. I want you to look at it and write some observations. And then I want you to increase the population size to 50. Do not change the generation and do not change the allele frequency. All right, just change the population size to 50. Again, plot 5 times without clearing. Generate a graph and put it in here. And then do the same for population size 100 and 500. And then we'll do it for 5,000 as well. All right. So now you should have different size population and all the graphs on the right-hand side. I want you to compare these graphs and write your other observations down here before moving on to your conclusion. So after comparing your graphs, I want you to write your final conclusion down and compare it to the hypothesis that you made before. All right, pause the video and do that now. And let's look at the explanation bit. Now, having alleles being fixed can be very dangerous for a population. All right, you never want to have only one allele in a gene. And I want you to think about why. Now, in biology, we have something called biodiversity, right? We want a different variety of species on Earth. We don't want to decrease our number of animals or different types of animals. The same thing. For genetics, we want to maintain a genetic diversity for a gene. We never want to only have one allele for a particular gene. Think about why. Have a chat to a friend if you're working with a friend, and then I want you to type your answers down here, and we'll go over it later.